Hi, thanks for coming. I'm going to go ahead and start. I haven't heard the chime, but it is 4:15, so for time reasons, we'll just go ahead and get started. Today I'm going to tell you about some of the work I've been doing in bedbug genomics. There it is. All right, so we are currently living in the century of the city. And as of 2008, we found that more humans lived in urban areas than in rural areas. So if you look at this map, countries that have the dark green have a higher percentage of their population that live in urban areas. And then when you go more into that brown scale, those are ones that have their populations more in the rural populations, okay? So you can see in 2008, a pretty large proportion of individuals were living in these urban areas. And now as we move to, slightly more recent, 2020, you can see that more and more of these countries are moving into this dark green space of having all populations in urban areas. So these are really densely populated urban areas that can have a large impact on the ecosystems. And again, we're thinking century of the city. I'm showing you 2008, 2020. But in reality, cities have been around for a really long time in human history. In fact, the first cities were found in the Mesopotamian region around 10,000 BCE. So this is quite a long time ago. Obviously, these cities don't look like what we see now as a city, but we still have a high human population density in a human-modified landscape. One of the things with these cities is that they were created when hunter-gatherers were now able to pursue interests other than gathering food. So they can start getting into the arts, architecture, all of those. So they're very drastically altering the environment. And when you alter the environment, you're going to have some consequences for organisms that live within that region. So a couple of really high population density animals that you're going to see in a city are German cockroaches and bed bugs. And quite a number of studies have already looked into the pressures that urbanization has on things like selection, gene flow, and genetic drift. However, there's still widely unexplored areas within this field. And today I'm going to be talking about bed bugs in particular. So bed bugs have, the ones that we know today, actually started on bats. And they did a host jump about 220,000 years ago from bats to humans. And this data was collected with mitochondrial genes. And what they found in this kind of mitochondrial genome is that the bat lineage of bed bugs have significantly higher genetic diversity than those in the human lineage, and there's population structure based on the localities where they're collected. And you can see with the human lineage up on top that there's really not a whole lot of genetic variation, and that variation is shared regardless of where you sample these individuals. And this is consistent with both mitochondrial DNA and with microsatellite DNA. So lots of studies are showing the same pattern of population structure and genetic diversity. Bed bugs are a human pest. They're a human health pest of a bit of concern. They are obligatory hematophagous insects. Basically, that means that they're blood feeding, and these are actually blood feeding at all life stages. So you're always going to have that issue. In the past, in the early 1900s, we used DDT to try to eliminate infestations, and we had a near global infestation reduction. So we ended up seeing that there were almost no bed bugs for a period of time. However, in recent years, there have been studies that found insecticide resistance, And then on top of that, we have human global travel. So between this insecticide resistance and global travel, we now have seen this resurgence of bed bugs globally. Now, the bat bed bugs are found only in Europe, but with the human-associated lineage, we're actually finding the bed bugs globally distributed. They're found in temperate regions across the entire globe. You're not going to find them as much in equatorial regions, but we do have several identification units where they did find this species. Unfortunately, the equatorial regions are not devoid of bed bugs. It's just a different species that you find there. All right, so with this information, we wanted to ask, do genome-wide patterns of human-feeding lineage bed bugs mirror human demography, given their close association with humans? To do this, we sampled individuals from the Czech Republic, and we collected from 6 locales. 3 of the locales were in human dwellings, getting the human-feeding lineage bed bugs. And 3 we collected in caves on bats, so we could look at the bat-feeding lineage. Okay. Next, we sequenced whole genomes of these individuals on Illumina HiSeq, and we aligned these sequences to our draft chromosome-level genome that we have in our lab. And finally, to estimate demography patterns, we estimated pi to G minus D, the site frequency spectrum, and changes in effective population size. Next, we did demographic modeling using Hudson's MS. It's a slightly older program, kind of older than some of the people in the room. It's back from 2002. It's a really simple, great tool that we can use to either model no changes in population demography. We can also do more advanced modeling for like a single population or a single lineage, where we do things like modeling bottlenecks and expansions through time. So it's got a lot of power to detect what's happening in a population through time. Okay, when we identified the amount of genetic diversity in these 2 lineages, on the far side, left and right eludes me. Okay, so on the very far side, we have both the bat and human lineage together, so that's where you can see all. And the overall mean pi is actually quite low, and this is because bed bugs have a pretty significant inbreeding level. But as you move down the graph, we can see that our bat lineage actually has higher genetic variation than the human lineage. And again, this is genome-wide estimates of genetic diversity. So it's following the same pattern as what we saw in the mitochondrial DNA and in the microsatellite DNA. So this is like a nice sanity check that the genome-wide is still going to have those same patterns. Then when we want to look at how this variation is partitioned, we ran a PCA. And what we have here in circles, they're very small circles, but they're there. That's going to be based on the bat lineage, and the triangles are based on our human lineage. And each color represents a sampled locale. And what we find is in the bat lineage, each locale actually falls into its own cluster, even though the clusters still have lots of genetic variation within them. So there's high variance within it, but they're still clustering together kind of vaguely. However, the human lineage populations, they all form one cluster, and they're very tightly grouped, which means that there's not a whole lot of variation between those individuals. They're more genetically related than the bat populations are across. So next, when we had our site frequency spectrum, we ran a stairway plot analysis. So here we have, in the light pink is our human feeding lineage, in the dark is the bat feeding lineage. And we go backwards in time, so closer to me is very old, and as you move across the screen, you're gonna get recent time. And what we find is around 40,000-ish years ago, and around the last glacial maximum, both lineages experienced a population decline. And this decline is actually really consistent with a number of other insects in temperate regions. And this has to do with that kind of bottleneck effect of coming into regions that were previously covered by glaciers, right? Then when we look at more recent years, you can see that that bat lineage continues to decline, whereas the human lineage exponentially increases. And likely the bat lineage decrease is going to be due to that loss of habitat that we see with bats in general, and the fact that they're staying in Europe where the human lineage is now able to go globally. We might expect something like that to happen. Next, we measured Tajima's D, and this basically tells you how the variation is partitioned across the genome. When we look at bedbugs, both bat and human lineage, we do see that there's a skew towards positive Tajima's D, and this is indicative of a bottleneck, which shouldn't be surprising given the last slide showed that really big decline in effective population size. Then when we look at our bat and human lineage, it's not going to be just because of one or the other. So even though the human lineage has recently experienced a very high increase in effective population, there's still going to be that signature of the bottleneck across the genome. I'm gonna blow through the rest really super quick. Okay, so just to let you know, Tajima's D is the measure that we use for goodness of fit test to find our best fitting model. And when we found our best fitting model, we set 40,000 years as our time of our bottleneck based on our previous data that showed that bottleneck at that time point. And we set our expansion, our current population expanded to 2.25 million. And that, again, is based on our data from our frequency plot. And when we ran a bunch of our simulations, we find that the bottleneck population dropped all the way down to 225,000 individuals. Still pretty high, but then it got a magnitude higher in recent years. And the time of that expansion was 5 to 20,000 years ago. And here's where we get super excited. That 40,000 years ago where we got the bottleneck, not only was it during the last glacial maximum, but it's also the time when humans entered Europe and were living in caves. Okay, so this is modern humans entering caves in Europe 40,000 years ago. And humans experienced a bottleneck during this time. Then 5 to 20,000 years ago, we see humans starting to build those those ancient cities, and they're starting to move around and get their higher population size, which is now when we see our bed bugs starting to take off. And then today, we're living in these vast cities globally, and we just have a massive bed bug population. So with that, I'd like to thank all of my collaborators and funding sources, and I have no time for questions. Thank you.
Okay, thank you, Dr. Miles. That was a really cool talk. I had bed bugs in my apartment the first month of the pandemic, and treating bed bugs is not considered essential work, so we became very close. Very cool to learn about that species that I'm really good friends with. My name is Danny Jackson. I'm a postdoc at the University of Arizona in Sabrina McHugh's lab, and I'm going to share with you work that we're doing on the genomic response of Darwin's finches to a novel parasite. This is work we're doing in collaboration with Leo Campagna and with Jesse Rick. And this fits into the really broad context of how hosts are responding to novel parasites. Here I'm also showing this photo of Peruvian pelicans, which very recently underwent a really big population decline as a result of avian influenza. So this is an increasing global problem, largely due to human impacts and the ways that we're changing connectivity and ecosystems around the world. We have coronavirus and white nose syndrome in bats. And most of what we know comes from microparasites, but what I'm going to be talking about today is macroparasites. So we went into this with a little bit of unknowns about what we might find. And one of the potential outcomes that a species might experience when they are exposed to a novel parasite is, unfortunately, extinction, as we know from Hawaiian honeycreepers from avian malaria. But species will also sometimes evolve resistance or tolerance to this new parasite in their ecosystem. But between these two far ends of the spectrum, there's a lot of intermediate opportunities for really complex effects on the life history of organisms and ecosystem level changes, because parasites are a really strong and novel selective pressure that can really change and reshape biological processes. The system we're looking at today is Darwin finches. We're looking at these 3 species to the left. They live on the Galapagos Islands. And then we're also thinking about the avian vampire fly, which is native to South and Central America. The adults live and feed on fruit, which is likely how they got to the Galapagos in the first place. But in 1998 was the first record of the larval form in the nests of birds, which is where they parasitize baby nestlings. In the top left, you can see some of the larvae sucking the blood out of that baby, and they have these really characteristic damage to their nares that will persist into adulthood. So when we're thinking about how these species might evolve in response to this selective pressure, based on field data, we know that parasitized nestlings have lower hemoglobin levels, they're overall smaller, and they have lesions on their face and on their body. And then those beak deformations again persist to adulthood. So we were looking for evidence of selection, and these all formed some of our hypotheses that we might observe. If there's selection, we might observe selection on something to do with blood, something to do with anything to do with like skin integument and cell development. And it is a very bad situation. On this plot, you see prevalence on the x-axis and costs on the y. So the Galapagos hosts are in gray, Central and South American hosts, which have co-evolved with the parasite for millennia, are in the red dots, and the Galapagos hosts all have a really, really high prevalence. So pretty much all species on the Galapagos have really high prevalence. The Darwin's finches have about 100% prevalence on the large islands. We find this in almost all of the nests, and it results in a really high cost. So that top right corner is where the Darwin's finches live. 50% reduction in survival of fledglings. And then models also suggest that if nothing changes, this will likely lead to the extinction of some species. But one of the things that may change is the evolution of our hosts. These 3 species are the ones that we looked at, in part because they spanned 3 different genera of Darwin's finches, and in part because we also had access to genomic data from before the introduction of the parasite. And throughout the talk, I'll use these little icons made by Charlotte Holden to refer to them. But our central questions are, do we see genomic evolution on such a short time scale? Again, this selective pressure only occurred first in the islands in 1998, so really short time frames. Do we see the same patterns across species? And then do we also see biological functions that emerge as candidate processes associated with host evolution in response to this parasite? This is our sampling scheme. We used samples from my advisor in 2019, Dr. Sabrina McHugh, and compared those to whole genome sequence data that were collected by Peter and Rosemary Grant and then sequenced for the 2 papers by Lema Shani et al. In 2015 and 2016. And so we have this pre and post sampling design, and we did about 9x whole genome sequencing of these. And statistically, we did direct comparisons of pre and post with FST and DXY, and then we also did a set of analyses that require that you only look at a single population. And from these, we developed candidate gene lists that were unique to the post population. So if evidence of selection existed before the introduction of the parasite, we assumed that that is doesn't need to be included in our candidate gene list. And we used a gene cutoff of 0.1%. Threw those candidate gene lists into Panther and ran statistical overrepresentation tests and then visualized it in R. And brief note on our methods, obviously we're using genetic data from 2 different batches. We followed a lot of the recommendations from these 2 papers, which are incredibly helpful if you're doing this sort of work, really amazing supplements with a lot of annotated code. But we took a lot of care to make sure that we're observing the actual process in our data and not just a bioinformatic artifact. And our species cluster, which is great, so we didn't mess anything up. And in FST scans across the genome, we see really low FST on average, but some peaks coming out as potential candidate regions. And again, this is just one of the many statistics we looked at. But then when we look at the gene list that we developed, in comparing the 3 species, a lot of the genes did not overlap between the 3, but a handful did, 5, 6, and 6 between each species pair. And these were identified by different programs sometimes. So some of them were both identified by FST or by DXY or a mix of the 2. There's a lot of overlap between those 2, and then a lot of overlap between SWEDE and Tajima's D. And within species, comparing whether or not our programs are coming out with the same gene, quite a few genes are supported by multiple programs, but quite a few genes are also not. And none are confirmed by all of our approaches. But when we focus in on these overlapping sections of that Venn diagram between species and just look at candidate genes involved in selection in 2 of the 3 species that we're looking at, so some replicates here, we see evidence of a epigenetic response, which is a really broad category, but one that we definitely would assume could be in the first wave of like an early selective response. Then we also see multiple categories of wound healing and tissue development, so like blood coagulation, and then also genes involved in an immune response. And then when we break this out to look at individual species, we continue to see those same 3 categories coming out, with epigenetic responses across the 3, and wound healing, again, across all 3 species, and then immune responses in 2 of the 3 species. So in conclusion, we are seeing potential parallel evolution in response to this introduced parasite. It's a really short time frame, and I think that proposes potentially some optimism for these species, that something is changing in the allele frequencies in their populations already, even though this situation is pretty bad. And yeah, wound healing, immune response, and epigenetics. So next steps are to start kind of refining our gene lists, looking at some machine learning methods for time series genomic data. And with that, would love to thank all of our collaborators. A lot of people have given their time and efforts to this work, and we'll take questions. Yeah, in the back. Oh, that's you, yeah.
Is there potential for a coevolutionary response with kind of them adapting their immune system and then these flies becoming adapted to better evade that? And I guess another follow-up question is that, is there active gene flow onto the Galapagos with these flies, or is that population relatively isolated?
Yeah, it's pretty isolated. I don't know that anybody's looked for gene flow. But the population is so abundant on the islands that I think that they're doing pretty fine on their own. And I think the 2 species entering into a coevolutionary arms race would be one of the many ideal situations rather than potential for extinction. Again, we are on such a short time frame, so this is like 3.5 to 7 generations of these birds that detecting selection like this, I don't think that it's likely that the selective response of the birds is created a selective environment that would have an impact on the flies yet, but definitely down the line, potentially. Yeah, great question, thank you. I really sped through that, so we have like 5 minutes left. Yeah.
Are you thinking about doing potential like differential gene expression analyses? Because on such short time frames, you can see more like transcriptional variants as opposed to, I mean, I know you can't do DE between your time series, but kind of maybe looking between susceptible and non-susceptible populations.
Yeah, totally. So the question was if we're looking potentially at doing gene expression studies next, and I think definitely the evidence that that transcriptome responses might be coming out as selected upon in this is a great next step. It kind of forms the foundation of this, but a lot of these findings are very recent, so we are thinking about potential next steps, including sequencing just the candidate genes and looking at those across time, including from some more recent samples from the last field season too. Yeah, thank you. Yeah.
That's really fascinating. Do you potentially use some of those hosts from the native range of the fly and co-evolving with it, kind of get a sense of like what they look like at some of those key genes?
Yeah, that's a great question. The question was if we could look at these key genes in the native hosts on South America, and I think that's a great question. It's not one that I thought about, so thank you. Okay. In the back.
What do you intend to do with the machine learning? How will it help in detecting selective signatures, or like how? I was just like, how will machine learning help to identify these signals of selection?
Yeah. So the paper that I referenced briefly, I'm still learning all of the details of it, but I know that it uses SLIM to simulate patterns of selection and then uses that to train an AI to detect similar selection in your empirical data. So we'll be looking at the genomes here. Again, this is such a short time frame that we were a little bit surprised to see something like this come out with FST and DXY. So those stats seem not super designed for a situation like this where it is a short time frame, it's the same population, and it's time series data. And yeah, we might expect different patterns of selection there. Yeah. Thank you. Yeah.
In line with the question earlier, you might want to look at the work of Charles Perrine at the University of Tulsa, and he works on cliff swallows.
Mm-hmm.
They're highly parasitized by a swallow bug, which is it's essentially a bed bug of cliff swallows. And the work that was done in the 80s by him showed this dramatic effect of this parasitism, and within a handful of generations, they lost that. So they have evolved strategies to evade the detrimental effects of these parasites, and yet the parasites are still there in densities of over 1 million per cliff-wall colony.
Cool.
That might be work you might want to look up. There's no genetic data into that there, but there is the traits of the birds in response to these parasites, which might be interesting.
Yeah, thank you. Yep.
It's maybe a really naive question, but I was wondering like how sensitive it is to the number of birds that you sequence in your results, and like if you expect to observe like if obviously you could always imagine to increase the sample size, but like how sensitive is it to that sample size?
Mm-hmm. Yeah, all of these stats are sensitive to sample size. There's a recent paper, it might be from one of the batch effect papers that I shared, where they have a plot of sequencing depth by individuals. That's a really helpful tool for identifying like what sample sizes might be effective at answering your question. And since we're looking at like small windows, yeah, definitely it is having an impact. We're also considering expanding into several of the other Darwin finch species too, to kind of increase the comparisons and develop this a little further. Thank you. Okay.
But yeah, I'm going to be talking about a study I've been doing on genome and transcriptome-applied analysis of advanced age in these genetically diverse populations of Saccharomyces cerevisiae with my PI, Dr. Molly Burke, and our collaborator, Dr. Felipe Barreto at Oregon State. And so, as I'm sure you've all observed, aging is a trait that is pretty much universally conserved across the domains of life. But from deep sea sponges that can live for tens of thousands of years to the mayfly, which has an adult lifespan of only a day, there's an incredible amount of variation in kind of the rate and timing of aging, both within species and between them. And so I'm interested in investigating the genes and gene regulatory pathways that are contributing to this variation and can be acted on by natural selection. And so there's a lot of ways we can study genes involved in aging. One really powerful way is called a GWAS, or genome-wide association study. This is a genomics session, so I'm sure many of you are familiar with that. But if you're not, it's essentially taking two groups, one group that has your trait of interest, in this case advanced age, and one group that doesn't, and comparing the average genomes across these groups. And so I have a Manhattan plot on the bottom of that far panel, And where we see those peaks in the Manhattan plot are regions of interest in the genome. So, the x-axis usually represents a position along the genome, and y-axis is a measure of significance. So these studies are really popular in medical literature. There's a lot of studies on aging in humans, but when we're trying to look at aging from an evolutionary perspective, there's a lot that we can gain from looking at model organisms. And so that's where my lab comes in. We study the yeast Saccharomyces cerevisiae, and specifically, we study populations of yeast that harbor a lot of genetic variation. So I study this 4S population, which is a synthetic population that we created by outcrossing for geographically and genetically diverse strains of yeast. And so using yeast, we can get much larger population sizes. It's much easier to control for a lot of confounding variables, and it's quite a bit easier for us to combine genomic and transcriptomic sequencing, for example, which is really useful when you're looking at complex traits. And so Saccharomyces cerevisiae is also a pretty historically used system for aging research. There are two ways that we can measure aging in yeast. One is chronological lifespan, and so that's just the amount of time that a yeast cell has survived without dividing. And the second is replicative lifespan, or the number of times that a yeast cell is capable of dividing asexually. This is a photo I took of a yeast cell that is currently undergoing asexual division. It's a little bit hard to see up on the screen, but on the bottom, there is a mother cell that has a lot of those little green rings. Those are called bud scars. And then at the top, there's a bud that's coming off of that mother cell that is kind of the newly synthesized cell. And so during each division, a bud scar forms at that site of division. And so we can stain these bud scars and use them to count, we can fluorescently stain them, and then count the number of bud scars on a cell. So that we know that old cells have a lot of bud scars, and young cells, newly synthesized cells, will have only one. And so this is really useful experimentally, where we can actually sort cells using fluorescence-activated cell sorting into young and old groups. And so I'll be talking about replicative age for the rest of my talk. And so my aims are to detect SNPs associated with the ability to survive to advanced age. And the genes associated with those. Detect changes in gene expression associated with survival to advanced age. And also investigate regulatory mechanisms that might be suggested by this combined data. And so this is a quick overview of my experimental design. I will essentially create 12 replicate populations. I can age them for long enough to establish a gradient of cell ages within each population. And then I can stain them, and importantly, I sort them with a paired design. So I will sort each population into a young cohort and an old cohort that I'll be comparing throughout the experiment. I'm not going to get into my kind of background phenotyping on these, but we do have evidence that that process works. And then after sorting the cells, we grow them up to log phase growth to standardize for the transcriptomic data. And so essentially what I'm doing is kind of a single cycle of a selection experiment, where I'm actually looking at the offspring of the aged cells or the young cells. And so from there, I go on and do my genome sequencing and my transcriptomic sequencing. So I'm going to start out by talking about my DNA sequencing results. And so you can see here there's a couple peaks that are sort of peeking out, but there's also a lot going on. So I started out by generating my significance threshold, which is that blue line. And so this is another Manhattan plot, kind of like the one we looked at the beginning. And this blue line is essentially separating out the top 1% of most significant SNPs, or single nucleotide polymorphisms. There were 669 of these SNPs, which were within 204 distinct genes. And so I did a gene ontology analysis of these, which tended to return genes involved in glycosylation, genes involved with the cell periphery, and that sort of thing. Glycosylation is also often involved in cell targeting, so that was kind of interesting at first. But when you look at these genes all together, a lot of them are involved in cell wall remodeling, which is a phenotype that has support for being related to replicative aging in the literature, but it's also something that's probably more specific to yeast rather than more of a conserved phenomenon. So that's not really what I want to focus on. But if I zoom in on some of these specific peaks, there are a few other interesting features of this data that I wanted to point out. And so I'm not going to get into all of it, but I do want to point out 3 genes in particular. So the TOR2 gene is in that chromosome 11 peak. The FRE3 gene is in the chromosome 15 peak. And then the RFA3 gene is in the chromosome 10 peak. And so these are 3 allele frequency plots that I've just thrown up on the board. On the left-hand side of each plot, I have the frequencies for the young populations. The right-hand side are the frequencies for the old populations. And those lines show the paired nature of the data. And so you can see that these peaks are being driven by relatively small changes in allele frequency, although that's not super surprising. We only had 1 cycle of selection. It was relatively weak selection. But overall, these peaks are being driven by extremely consistent changes in allele frequency across populations. In most cases, nearly every single replicate has a very similar kind of change in allele frequencies between the young and the old populations. And specifically, I pointed out these 3 genes because they're all ones that have ties to aging in literature. The TOR2 gene is involved in TOR signaling, which is well known or has been implicated in aging in both yeast and also its ortholog mTOR in humans. FRE3 is involved in iron homeostasis, and metal ion homeostasis is associated with oxidative stress. And finally, RFA3 is a replication protein. DNA replication stress is also associated with aging response. And that protein in particular is involved in DNA repair, replication, recombination, and also has some functions associated with telomere protection and maintenance. So I want to put a star on that gene too, because it's going to turn up again later. But in the meantime, I want to talk a little bit about my RNA sequencing results. Just to orient you to this heat map here, all of my old replicates are on the right side, and all of my young are on the left side. And then up at the top, there's kind of that rainbow bar that might be kind of hard to see, but that indicates the paired nature of my data. So the red is pair 1, blue is pair 2, green is pair 3, and so forth. And these patterns are a little bit more evident when you really stare at those pairings, but I also have that bar on the far right that shows the log fold change. And so blue in that bar indicates a decrease in expression in the old, and yellow indicates an increase in expression in the old. With a p-value of 0.1, 41 genes were implicated in the RNA sequencing data. 26 of those had a p-value less than 0.05. Not a lot of genes, right? But remember, we're looking at the offspring of our young and our old cells rather than the young and the old cells themselves. So that wasn't super surprising to us. Also not surprising that we didn't really see any significant go terms because there aren't a lot of genes in general. But we did see that RFA3 gene popping up once again. And that's actually the only gene that was implicated in both my DNA and RNA sequencing, which I was a little bit surprised by. And even though there were no significant go terms, I did just pull out a couple functions that seem to be shared between some of these genes, including a lot of downstream modifications, some chromatin remodeling, DNA repair, and a lot of these functions, actually I think every single one of these functions, were also shared by significant DNA sequencing genes. And so now that we've looked a little bit at both the DNA and the RNA sequencing data, I also want to try to see what conclusions I can draw from putting this data together that I might not be able to find from either data set individually. And this is still a little bit preliminary. I'm still kind of digging through this data. But specifically, I'm looking for evidence of cis-regulatory regions that are immediately upstream of my implicated genes. And by that, I mean within 5 kilobases that might kind of be impacting the regulation of aging. And so this is actually the same Manhattan plot I showed earlier, but this time I've colored all of the genes that were implicated in RNA sequencing in red. And so it's a little hard to see up there, but there is that RFA3 gene, which was the only gene that was implicated in both forms of sequencing. So far, just looking at this, there's not a lot of evidence of that cis-regulatory interaction. I dug a little bit deeper, though, and I can give more details about how I did this if you're interested later. But essentially, I pulled out the types of variants that are associated with each of those significant SNPs and those 204 significant genes. A lot of these were non-synonymous variants, so it's possible that some of these are altering protein structure without impacting expression, and that's why we're not seeing this overlap. But a lot of them also occurred in these upstream gene variants, and actually that was the largest category. There were over 60 genes that only included an upstream gene variant. So it seems like these are probably doing something. But given the lack of overlap between our DNA sequencing and RNA sequencing data, we don't think it's likely that it's a cis-regulatory interaction. There's likely something else going on. And so I'm running out of time, so I just want to wrap it up. But the RFA3 gene does appear to be an important gene, and so that would be interesting to investigate further. But there's not a lot of overlap overall between my DNA and RNA sequencing data, and we're not finding evidence for cis-regulatory interactions. And there's a lot of future directions I'm interested in, some of which I'll get to, and some of which I will probably not. But I also want to take the time to thank my lab and thank our funding sources, and thank you for listening. Maybe I have time for one question? Yeah.
Yeah, so I have a question. Based on the SNPs you showed us, do you look at the distribution of like the allele frequency distribution of your candidate SNPs? Is it the case that they're skewing to like things that are just at higher frequency in the population, or are you seeing any evidence of shifts in like potentially things starting off at a lower frequency that maybe might have more of a signal?
Oh, like, yeah, I haven't had a chance to really dig into the ancestral data yet. I've really only been looking at like my young versus old. So it'd be really interesting to look at that. But yeah, I want to do the haplotype tracking to try to see like what genomic backgrounds some of these SNPs may be coming from. But yeah, I just haven't had a chance to do that yet. Thank you.